right, hi everyone. It's my pleasure to introduce Deren Eaton, who is an associate research scientist at Yale University, and in July begins a tenure track faculty position at Columbia. Prior to that, Deren did his undergrad at University of Minnesota, where he worked with Janine Cavender-Barris, and then PhD at University of Chicago, where his advisor was Rick Reed, who's the curator of botany at the Field Museum. And then he went on to a postdoc with Mike Donahue at Yale before beginning his current position. Deren's a botanist. He's worked quite a bit throughout China, and as well as more recently southern Mexico. And he'll tell you all about the biology today. But what I wanted to highlight in the introduction is that in addition to all the science that you're seeing here, Deren has developed a lot of bioinformatic tools that have been very useful for other people. So all the RAD-seq data here, Deren basically, as a first or second year grad student, started writing this program, PyRad, that ended up now being one of the most popular programs to analyze this kind of data. I use it all the time, and has also been kind of a pioneer in some open, reproducible science. So, for example, his recent evolution paper, you can actually go on to his website and see step-by-step tutorials to reproduce every single analysis in the paper, which is really helpful. So anyway, let Deren get on to the science.
Thanks, Ben. Yeah, thanks for coming, everyone. Today, I'm going to tell you a little bit about my research. So very broadly, I study plant phylogenetics, and I'm interested in the diversification of flowering plants, understanding why some lineages are more diverse than others, and why some regions of the globe harbor more diversity of plants than others. And sort of in a more narrow focus, I'm particularly interested in the role of reproductive evolution and reproductive interactions in driving those patterns. So the evolution of flowers and of interactions between plants and pollinators, and between plants with each other with respect to their reproductive interactions, including processes such as gene flow within and between species. So I'm going to talk about a number of examples working with different plant groups in which sort of share that common theme today. And then another sort of common theme across all these studies is the use of large-scale comparative genomic data sets in the form of RAD-seq data. So I'll start by giving a little background about the genomic data in particular, and then really focus on the biology. So I think this will be familiar to a lot of people, the idea that for a long time there was limits on the type of genetic data that we could gather and use for phylogenetics. In many ways, we were limited in the use of Sanger sequence data to sequencing a small number of genes, often across a large number of taxa. But those gene trees were sometimes poorly resolved, and we had little statistical power, if we only sampled a few of them, to explain conflicts between these different gene trees. Alternatively, we could sequence using, say, full genome sequencing approaches a small number of taxa and have a large number of gene trees in which we could use statistical methods to explain discordance among these gene trees. But due to the high cost of it, we can only do this for a small number of taxa. So we can't really gain insights across whole entire clades using that approach. Ideally, we want to be able to connect sort of one continuous data type from the scale of populations and individuals to larger and deeper clades. And we're still kind of a ways from having that kind of data, but increasingly we're getting closer to it. And I think a real goal of having that kind of data is being able to connect variation within and between populations up to the level of explaining, say, differences in lineage diversification rates and patterns of trait evolution within larger clades. The limitations we have in generating sufficient genome scale data for doing this is largely driven by limitations of the technology. So when we use shotgun-type sequencing approaches, we're more or less distributing randomly reads across the entire genome, such that we're putting a lot of effort into sequencing regions that aren't really going to be used in downstream analyses. They're not easy to compare and map across different taxa. Newer methods, so-called reduced representation genomic methods, are a lot more efficient in that they essentially reduce and target the sequencing just to certain parts of the genome before you actually do the sequencing. And there's several types of methods for doing this, one of which is called RAD-seq, or restriction site associated DNA sequencing. And this targets sequencing to regions that are adjacent to restriction enzyme recognition sites in the genome. To the extent that these recognition sites are conserved across all of the individuals or species that you're sampling, you expect to attain the downstream nearby sequence from all of those as a marker for all of those taxa. Doing this, you can get really high coverage data for a subset of the genome, still possibly tens of thousands of regions, and do so across thousands of individuals for relatively cheaply. RADseq was originally sort of pioneered as a method for studying relatively shallow scale relationships, but increasingly it's being applied to deeper scale questions, and it's doing so quite successfully. One problem, though, that you run into is increasingly sparse matrices, which have a lot of missing data. So you end up getting markers that aren't sequenced for all of your individuals. And to some extent, you find increased missing data between more distantly related individuals because there's a opportunity for greater mutations to arise between them that can disrupt these restriction recognition sites. Yet this method has still been applied to scales stretching to many tens of millions of years successfully. So for relatively deep phylogenetic scales, it still seems to work quite well. And I've been really involved in trying to make this method work better for these deeper evolutionary timescales by developing this software PyRAD, which it can in many ways assemble more complete data sets by accounting for indel variation when identifying homology between sequences, which some of the other software available doesn't do because it was really focused on more shallow scales. So you can recover much more complete and informative data sets. We now have a new version, a sort of complete rewrite of this software that's available now called iPyRAD, which has a lot of cool new interactive features. It's much faster than the older version and has a lot of downstream analysis tools built into it. So if you're using RADseq data, I encourage you to check that out. So now that we can gather these large-scale comparative data sets rather cheaply and efficiently, what kind of questions can we ask or should we ask? And what have we learned over, say, the last 5 years or so that these methods have become available? I think one of the really big themes that has become really apparent from this type of data is the extent to which historical hybridization is occurring between lineages, which in many ways we were not quite so aware of. So I've sort of broken the talk into 4 sections which are variously related to using genomic data and understanding gene flow better in different ways. So this first section I'm going to really talk about detecting gene flow. I think the study of hybridization and admixture really took off around 2010 with the publication of this sort of seminal paper showing evidence of introgression between humans and Neanderthals sometime in several tens of thousands of years ago. And in addition to this really exciting result, the paper was groundbreaking also for introducing a really simple and elegant method for detecting admixture, giving a genome-wide alignment for at least 4 taxa. The way this test works is you're looking for sites throughout the genome that give a discordant pattern, allele pattern, relative to your hypothesized species tree. In this case, we're looking for sites throughout the genome in which you have a derived allele, here shown as B, relative to your outgroup, and where that derived allele is shared with one but not the other of 2 sister species. So these patterns are discordant with respect to the expected concordant site patterns, such as BBBA. You expect that these 2 patterns should arise with equal frequency due to the sorting of ancestral polymorphisms on this tree. But to the extent you have a pattern of introgression between P3 and either P1 or P2, you would get an asymmetry in their occurrence, an overabundance of one pattern or another. And the D statistic is simply a measurement of that asymmetry. So D that deviates significantly from 0, meaning you have more occurrence of one of these patterns than the other, is suggestive that there's been admixture between these 2 lineages. I became really interested in applying this test to RADseq data sets that I gathered for a number of plant species to test hypotheses about hybridization between species. And I found really quickly that you can run into some problems when you apply it to deeper evolutionary timescales. In particular, it can lead you to really overestimate the frequency at which gene flow has occurred between different species. And this arises particularly in the case when you have multiple potential species that could be the introgressive donors in a scenario of hybridization. So consider the case where taxon C has introgressed into taxon B, and taxon C is part of a clade that includes 2 other closely related species, D and E. Well, in this case, if we apply the 4 taxon D statistic and we substitute each of those 3 different species in as the P3 sort of introgressive donor taxon, we find a significant result in all 3 of those tests. It's of decreasing significance in each of these, but we can't actually distinguish whether each of those species introgressed into B independently or whether only one of them did. What's happening here is that introgression that occurs from C into B is introgressing alleles that are unique to the C branch, but also alleles that arose uniquely on this lineage along the C to D and the E to C to D branch. And that's why you're getting a significant signal of admixture from all three of them. You need to tease apart the signal of their shared ancestry from that of the uniquely introgressed alleles in taxon C. So I developed an extension to the D-statistic test that's aimed at trying to get around this to more accurately detect where the actual source of introgression is, which branch on the tree. The way we do this, and there's been other extensions to this test developed since then as well, is that we try to separate out alleles that are uniquely introgressed from one individual branch from alleles that arose in the ancestor of that branch. So we take that P3 lineage and we now split it into two, and we sort of move our way up that P3 clade, measuring the left branch versus the right branch at a time, and asking whether the alleles are derived in both of those two left and right branches, suggesting that that derived allele is present in their ancestor, or whether it's uniquely derived just to the lineage on the left or just the lineage on the right. And this gives us three different D-statistics then, measuring the ancestral introgressed alleles and those that are unique to each lineage. So I'll show you an example of this, applying it to an empirical data set, which I think makes really clear its importance. And there's probably no better group to apply this to than oak trees, which are sort of a very notorious group for hybridization, where it's thought very widely that species are sort of going crazy and hybridizing in a lot of various ways. So this is work that I've done in recent years in collaboration with Janine Cavenderbears at the University of Minnesota, with Andrew Hipp at the Morton Arboretum, and with Antonio Gonzalez-Rodriguez at UNAM. In particular, we focused on one small clade of oak trees, the American live oaks, which is a group of 7 species, a monophyletic clade with a crown age of about 10 million years that's diversified in North America and Central America. It has a really interesting distribution, essentially forming a sort of ring-like complex around the Gulf of Mexico, with one species that's endemic to Cuba, another species that's endemic to the tip of Baja California. And they've diversified into a huge range of ecological habitats, from sort of tropical wet forest to dry forest, to frost-tolerant species in northern Texas, and this sort of desert endemic in Baja. So we wanted to better understand the phylogeny of this group and the extent to which species have exchanged genes in the areas where they're either in sympatry or in parapatry. So we sequenced the RADseq data set, sampling broadly across each of these species, 4 to 5 individuals across their range, and we recovered around 30 to 60,000 rad loci per individual. We inferred the phylogeny using a number of methods. I'm showing the RAxML maximum likelihood tree here. In general, we recovered really consistent results, but consistently with some species coming out as paraphyletic, which we think is a result of gene flow, which I'll explain. We find a deep split in the tree separating these 2 westernmost species from the other 5 easternmost species. So this includes Quercus fusiformis in Texas and Mexico, and Quercus brandigii in Baja. The other clade includes a southern clade that has the Central American oak species and Quercus sagariana in Cuba. And then there's a southeastern clade that includes 3 species that are broadly overlapping in Florida, and one that extends into the southeastern US quite a bit. Using structure, we find some evidence of admixture between these different lineages. And we wanted to sort of interrogate that more specifically, genome-wide, using these ABBA-BABA tests. We find a lot of evidence of a sort of complex history of this Cuban oak being a hybrid between the Central American and Florida clades, which we go into great detail about in the paper, but I'm not going to talk about in this talk. Instead, I want to focus on introgression across the deepest split in this tree. So this is spanning about 8 million years of divergence. And in this case, what we're testing is whether Quercus fusiformis, which occurs here in Texas and Mexico, whether it shares more alleles with this clade of these other 5 oaks relative to its sister taxon, Quercus brandigii. So all else being equal, we expect fusiformis and brandigii to be equally related to all these taxa if there's no gene flow. Alternatively, we might predict that we're going to detect a lot of gene flow into fusiformis. And when we apply the 4 taxon D statistic, what we find is significant evidence of admixture from all 5 of those taxa with Quercus fusiformis relative to brandigii. So this would suggest that all of these species have potentially hybridized to the way that many people often interpret the results of this test. Well, if we apply the partition D-statistic test, this extension to it that I developed, we find that the number of hybridization events is actually much fewer. So what we do here is now we're contrasting 3 potential donor lineages as potential donors into fusiformis. I'll refer to the clades by their colors here, because I think that'll be simpler. So I collapsed the 3 southeastern oak species into one clade shown in yellow, and then there's a green taxon in Cuba and a red taxon in Central America. So we can contrast, in this case, the yellow versus the green taxa as potential donors, introgressive donors into the blue taxon. And what we find is a significant signal of shared ancestral alleles shared by the yellow and green taxa that are introgressed into the blue taxon. But we only find uniquely introgressed alleles coming from the yellow taxon into the blue taxon. The green taxa doesn't have any alleles shared with the blue that aren't also shared with yellow, right? So this suggests that gene flow actually came from the yellow taxon in that case. Similarly, we can compare the yellow versus the red taxon, and we find the same case, only uniquely introgressed alleles coming from the yellow taxon. And we can compare green and red, but it's not really important because we know neither of those two have uniquely introgressed alleles relative to yellow. So we can infer then that the true source of introgression into Quercus fusiformis is likely the yellow clade, with these two taxa just showing a signature of admixture due to their shared ancestry with the yellow clade. Interestingly, so this is focused on Quercus fusiformis sampled in the northern part of its range in Texas. If we sample it in the southern part of its range in Mexico, we find the exact opposite pattern. Now when we perform these different contrasts between these clades, we find that only the red taxon actually shows uniquely introgressed alleles into fusiformis. So what we're finding actually is a really localized pattern of introgression just in the region where they occur in sympatry. And you might say, well, that seems kind of obvious. I mean, you found introgression in between the two taxa that are actually sympatric in the north and between the two taxa that are actually sympatric in the south. But I think it's important to note that we found this result by applying this 5 taxon test, but we really didn't have power to detect that using the 4 taxon test. In many cases, you might have multiple species co-occurring together that you don't know, obviously, that one's the true donor and another one isn't. So it's important to perform these different contrasts. It's also important to consider that we sampled all of the extant species in this clade, and we sampled them broadly across their range in performing these tests to find these different patterns. Consider the case where we hadn't sampled the southeastern clade and we'd perform this test in the north, or if we hadn't sampled the Central American clade and we'd perform this test in the south. In both cases, even performing these comparisons, we would have been left only with the result that the taxon that seems most likely to have introgressed into Quercus fusiformis is the Cuban oak. So we could have been completely misled based simply on the fact that we didn't sample the correct taxon. Of course, this is a problem with the fact that clades can go extinct, and so there could be some clades that we'll never be able to sample. But to the extent we're able to sample many clades, it sort of runs the risk of comparing only very distantly related organisms, asking whether they've introgressed, when in fact it could be a very far distant clade of one of these organisms that actually exchange genes in this relationship, and you're just picking up a sort of tangential signal of that. So the sort of take-home lesson is that this test and many other types of related tests for introgression and admixture are inherently a type of comparative test, and so it really depends on what you're comparing in the accuracy of your results that you're going to end up with. So gene flow seems to be quite common in these oaks, but it's by no means ubiquitous. And we find a lot of evidence of introgression between sympatric species, but we don't find it across the full range of all of these species. I think to the extent that we're finding that there's a lot of gene flow increasingly between different groups of lineages that we interrogate, I think we should think about what this means in terms of how we're quantifying biodiversity. Do we expect that we're going to increasingly collapse things that we previously described as being distinct and now say, well, maybe they're the same species if they're exchanging genes? Or as we apply more and more large genome scale data sets to understudied organisms, are we going to uncover lots of cryptic diversity and increasingly describe many new species? In flowering plants, it's thought that still maybe as much as 10 to 20% of flowering plants remain undescribed. And it's thought that a lot of these are probably tropical trees, which are just very difficult to study from a taxonomic perspective. I've been working recently on a group of tropical trees, collaborating with Sarah Fetterman, a graduate student at Yale, and with Doug Daly, who's a curator at the New York Botanic Garden. We've been working on a group called Canarium, which is in the Burseraceae. It's a relative of frankincense and myrrh. These are really large dioecious trees that produce large fruits, and they're a major food source for lemurs in Madagascar. And they're really widely distributed, a dominant tree in Madagascar in both the wet and dry forests. And this clade appears to have arrived in Madagascar only around less than 10 million years ago, and has really sort of diversified and become a really dominant tree in this landscape. Sarah and Doug recently completed a new monographic treatment of Canarium in Madagascar, which had some really kind of astounding results. The previous treatment of Canarium in Madagascar described one species in 1959 with 3 subspecies, and Daly and Fetterman recently described that there was 33 taxa. So that's a huge difference, obviously, that sort of requires explanation. So One explanation is that historically there were very few samples of Canarium from Madagascar. And sampling these trees is actually really difficult. It's difficult and dangerous to travel in many regions of Madagascar. And these plants are very tall, and so it's difficult to collect reproductive tissues that's required for identifications. And they're dioecious, which makes it even more difficult. So it's possible that simply better sampling has allowed us to identify many more species, or it could be that we're just very into oversplitting species these days. So we decided to test this new 33 species hypothesis using genomic data and sequencing many of these taxa. So we sequenced a GBS library for 48 individuals. We were able to sample 17 out of these 33 species. Many of them were described from very few specimens, and many of those were not collected in a way that we could get good enough DNA out of them. So we have multiple individuals from many of these taxa, but not from all of them. And we sequenced it to very high coverage. We recovered about 55,000 loci per sample, and a assembled data set with about 40% missing data. What we find sort of the deepest splits in this tree gives 3 major clades that seem to correspond really well morphologically to those 3 subspecies described by Lienhoutz in 1959. And we find some more structure and that fits well with some of these new names described by Daly and Fetterman, particularly among some microendemics in the north, which is known to be a biodiversity hotspot in Madagascar. We then find that there's a lot of names described within, new names described within these 2 major clades here shown in yellow and green. And this is a structure plot along the right side. And so there's two clades that are really widely distributed along the wet forests on the eastern side of Madagascar that are sort of co-distributed. And then there's this red taxon that's thought to be really widely distributed along the dry forests on the western coast. Well, when we actually look at all of these names as they've been described on this tree, these lines here are connecting taxa that were described to have the same names. Many of these, all of these new names ascribed to these taxa. We find that there's a huge amount of paraphyly of a lot of these names that were newly described, particularly between these 2 really widespread eastern clades. So it appears that many of these names are likely invalid, either some kind of poor identification, or what we think actually is probably that there's quite a bit of admixture between those 2 clades, that when these taxa are being described from few specimens was kind of hard to recognize as being perhaps part of a continuum of variation or being hybrids rather than being a unique taxon or population. So we find a fair bit of evidence using these ABBA-BABA tests and the partition tests, particularly between those 2 widespread eastern clades. And we think a lot of this taxonomic paraphyly came as a result of that. In the end, a new treatment, or at least for the species that we sampled, we think there's around 6 well-described, well-delimited species using statistical species delimitation approaches. So this is somewhat of a cautionary story for the expectation that perhaps we're going to continue to always discover and describe new species as we apply new genomic methods to poorly studied groups. In some cases, actually, the data might give the opposite conclusions. So the next study I want to talk to you about is kind of my main dissertation work, which is still unpublished, but hopes to be soon. So a lot of my PhD was focused on a group of flowering plants called Pedicularis, which is a widespread, really diverse group of herbaceous flowering plants in the parasitic plant family Orobanchaceae. There's about 600 species worldwide, distributed mainly in alpine regions as well as sort of circumboreal distributions of several species. But the vast majority of that diversity, more than 300 species, are endemic to just one small region on the eastern edge of the Tibetan Plateau. So there's a huge concentration of their diversity in this region, in this really high elevation habitats between 3,000 and 5,000 meters. And this region is known as a hotspot for temperate plant diversity, alpine plant diversity. The work I'll describe in this section was done in collaboration with Huang Shuangchuan, my collaborator at Wuhan University, and with my PhD advisor, Rick Ree. So in trying to sort of understand and investigate the extreme diversity of Pedicularis in this region, the first thing that's really striking about this group is their really extreme amount of floral diversity. You find not only a huge diversity of floral types, but a sort of extreme occurrence of convergence, a convergent evolution of similar floral types among very distant clades. And these floral variation that we see within these species has really important functional consequences. One hypothesis we have for what might be driving this extreme diversity of floral forms is interactions between species, particularly negative reproductive interactions in the form of interference. One reason we suspect this is that the environment imposes a number of constraints on reproduction. You have this very short growing season in these high elevation alpine habitats. So the plants more or less are trying to complete their life cycle in about 3 months. Their flowering times are about around 1.5 to 2 months long. So they're broadly overlapping. These 300 species in this region are broadly overlapping in their flowering times. They all are obligately pollinated by bumblebees. So they all share these generalist pollinators. And so there's this high potential, not only that they would compete for these limited pollinators, but also that they have to avoid interspecific pollination between each other. And they've evolved some floral traits that seem to really be functionally important in avoiding interspecific pollination. One of these traits is the galea, and this is a fusion of 2 upper petals of the corolla, which encloses the anthers, which you can see in this sort of transparent corolla here. So that's where the pollen is placed onto bees during visitation. And in many cases, the galea, several times independently, has evolved a sort of closed structure and an elongation where it becomes variously elongated and curved. And it's sort of like an elephant's beak structure. It acts as sort of a funnel to direct pollen placement onto really specific parts of bees' bodies. As you can see here, being placed on the ventral side or on the left side of the bee, versus these nectar-producing forms tend to have an open galea that places pollen on the dorsal side of the bees. Another interesting trait that has evolved multiple times, 3 examples here of 3 very distant relatives that all have evolved this really long, really elongate style and corolla tube. You can see in my hat here for scale. So this is not a sort of vegetative tissue, but this is floral tissue, actually, a fused corolla that stretches from the ovules at the base of the flower. The style will reach through this corolla tube all the way out through the tip of the galea. So this can sometimes be 10 or 15 centimeters long, and this is the length that the pollen tube has to grow when it lands on the stigma of the flower all the way down to reach the ovules of the plant. So one hypothesis for a functional significance of this trait that's evolved multiple times is that it provides this gametophytic gauntlet in which certain pollen tubes can outcompete others. In particular, species with really long styles tend to have larger pollen that grows faster and it grows a further distance than smaller pollen. So this is a way to, again, avoid interspecific pollination. So we have this extreme diversity of Pedicularis in this region and this extreme disparity of their floral forms. We wanted to ask, is it possible that competition and interference between species has in part contributed to their diversification by driving floral phenotypic divergence? And to the extent we find floral divergence occurring within species, is that contributing to speciation and isolation? Our first approach to look at this was sort of a community phylogenetic type approach, looking across a huge database of collections of Pedicularis in this region. And we looked across more than 200 communities that recorded which species were co-occurring with each other and co-flowering. Communities that had between 3 and 10 species that were co-occurring together. And we found that given a multivariate floral morphospace that we constructed, that species that co-occur tend to be distributed from more disparate parts of this floral morphospace than expected by chance. So that's consistent with a pattern of species at least sorting across the landscape into communities by a process of limiting similarity to avoid a competition. More interesting, though, is we would like to know what are the sort of detect evolutionary consequences of these species interactions rather than what might just be sorting across the landscape. And for that, what we're trying to detect is evidence of character displacement. So character displacement is a pattern where 2 species show different phenotypes when they occur in allopatry versus when they occur in sympatry. Due to competition for some limiting resource, they'll diverge into alternative resources. Well, in the case of Pedicularis, it's difficult to just ask whether one taxon has a different phenotype when it co-occurs with some other focal taxon, because any given species in this region is likely to co-occur with dozens of other species, and many of those different species may convergently fill the same floral niche. So there's a real difficulty in trying to identify one focal competitor. So what we did is we tried to come up with, rather than a simple one taxon character displacement test, a sort of community-based test for character displacement. And in addition to just focusing on phenotypic characters as a sort of selective factor, we also included phylogenetic information as a potential covariate, because it could be that closer relatives have a more detrimental effect in exchanging pollen. So to do this, we focused on one widespread and really common species of Pedicularis, Pedicularis cranulofa, which I collected this really broadly across northern Yunnan province and southwestern Sichuan province, where it co-occurs with several dozen other species. And across this range, we find a huge amount of floral variation within the species, which is part of the reason that we chose it. So you can see between these 2 neighboring populations, we have about a twofold difference in the length of the style of these flowers. So the variation within this species is really apparent. So our hypothesis then is that the variation that we observe between local communities of our focal taxon, Pedicularis granulosa, is a result of the local selective pressures that it experiences in the communities with which it occurs. So you could imagine population 1 occurs with a community of species that doesn't impose very strong selection for it to have a longer style, whereas community 2 co-occurs in a biotic community that puts very strong selection on it to have a longer style that might help it to avoid gametophytic competition. So what we're testing then is for an association between the phenotype of our focal population and something about their environment, in this case a biotic environment that they're experiencing. And if we do that, we need to control for the phylogenetic relationships of our focal populations, because some of these populations might simply appear really similar because they're very closely related, not because they experience similar environments. This is the idea of phylogenetic corrections explained by Felsenstein in '85. Well, it's also a few years later, Felsenstein described an alternative test which says, in the case that you have a lot of gene flow going on between different populations, a more appropriate test, rather than using a tree, is to use what's called a gene flow or a migration matrix. And this alternative model was described as a method for controlling for non-independence of populations that are exchanging genes. And so I'm going to explain how we decided to go with this approach instead, which is a somewhat novel application. So first, I'll explain the data. We sequenced RADseq data for 110 individuals across 15 populations of Pedicularis granulosa, so between 6 and 8 individuals per population. We sequenced it to fairly high depth and recovered about 70,000 loci per sample. We first inferred a tree, and we found that there's actually really strong evidence of population structure. So these individuals we sampled within each population almost always came out as reciprocally monophyletic, with only a little bit of what came out as apparent mixing or non-monophyly between different populations. Yet we do find evidence of what could be admixture between different populations, or at least a continuous amount of variation. And we find evidence of what could be a support for a strong pattern of gene flow using these ABBA-BABA tests. So when we look at, in particular, I think a good one to point out is this southernmost population, which comes out as a real kind of admixed population between the purple clade, which is the sort of first diverging clade in this lineage, and this yellow clade. And one way you could get this pattern, and this is what we find supported by the D statistics, is if this population was actually part of the yellow clade, but then received a lot of migrants from the purple clade, and that would sort of pull it outside of that yellow and red clade to appear in the position where it is now. Well, if you tried to apply phylogenetic models to model the trait evolution of this lineage, what would happen is it would appear like it's evolved independently for a long time on this really long branch, which would suggest that it's really actually poorly explained by the phenotypes of these other species, of the phenotype of their ancestor. Alternatively, if we apply this migration matrix approach, in this case we inferred a migration matrix using migrate n, and I'll focus on these 4 populations as an example of this. What we find alternatively is that in migrate we find strong evidence that the southernmost population is sort of a sink of gene flow. It's receiving a lot of migrants from its neighboring populations and not exchanging very much migration in return. Well, if we use this model to try to explain the phenotype of this southernmost population, this would suggest that its neighboring populations are actually a very good predictor of its phenotype. And so this is an argument for using an alternative model, such as a migration matrix, instead of a tree when trying to model trait evolution. So I'm sort of justifying that before I'm going to show you the example. So this method that Felsenstein described for trying to model trait evolution using a migration matrix is based on a quantitative genetic model described by Landy, which models a population mean as being drawn towards some phenotypic optimum, which can be different in every given population. But a population never quite reaches its optimum because it's always being drawn away from it a little bit by gene flow from its neighboring populations, which have their own mean phenotype. And then there's also some amount of drift going on. And so you can, given that you can, so you can think of this as these yellow dots being the local optima. And if there's a huge amount of gene flow, then all of the populations are homogenized, so they can't reach their local optimum. But if you crank up selection such that it overcomes gene flow, then they can diverge from that homogenized state and adapt to their local optima. So Felsenstein shows that if you can actually measure the amount of gene flow that's going on between these populations, then you can get at how much selection has actually occurred. So what you do is using your knowledge of gene flow and your local measurement of trait means that you're interested in, you can apply this eigen decomposition of the migration matrix to get independent transformed values for the trait means. So this is sort of like independent contrasts for traits on a tree. We then have some local optima, which we don't actually know what the local optima are in these populations, but there's something that we can measure, which is something that might be correlated with it, such as measurements of our local biotic community. In this case, we'll use phenotypic distance to the congeners in that community, or the phylogenetic distance to congeners in that community. We can ask how much our trait means diverge from or deviate from this local optimum, and that deviation should be correlated with the estimated variances from this transformation. So to recap, what we're going to focus on then is the focal style length in these 15 focal populations. And we measured a migration matrix using the RAD-seq data, and then we're fitting a model for these local optima, in this case using a Bayesian method to jointly estimate a coefficient for selection and drift, and then for a linear function that describes our local biotic variable as a function of our local optima. What we find when we try to use a phenotypic covariate in our model, whether it's nearest neighbor distance or mean distance, is that the phenotypic models are a really poor fit to the model. Alternatively, when we fit a phylogenetic model, in particular a nearest neighbor phylogenetic model, we find that it's actually a very good fit to the model. There's a strong selection coefficient and a negative slope to the fit, suggesting that when Pedicularis granulosa co-occurs with closer relatives, so a smaller phylogenetic distance, they tend to have a longer style. And this supports this sort of gametophytic arms race hypothesis, where potentially having a longer style length, a longer corolla tube, helps you avoid receiving gene flow from a close relative that might have a very detrimental effect in producing, say, unfit hybrids, or at least fertilizing your ovules. So what this means is we're detecting evidence then that there's interspecific interactions going on between these species that's driving intraspecific variation within the species that we see with respect to their style lengths. And the next question we ask is, what are the consequences of that, of this variation arising within the species in their reproductive traits? We tried to get at that through an experimental approach. So I took these two populations that were really close by each other and showed this largest difference in their style lengths, and I performed crosses, reciprocal crosses in both directions between them. And we then measured over different time series the rate of pollen tube growth using fluorescence microscopy. And what we found is that these species with the longer styles, which also have larger pollen, grow faster pollen tubes in both directions of the cross, and they reach the ovules first in both directions. So we would expect that there should be probably an asymmetry of gene flow that results from these differences that have arisen between these different populations. And indeed, from the migration matrix estimates, that's what we find evidence for 10 times higher migration rates from the long tube to the short tube than in the reverse. And so this is evidence that while not necessarily leading to isolation of these populations, these divergences that have arisen due to interspecific interactions are affecting at least the cohesiveness of these populations as a species by affecting the rates of gene flow among them, potentially. Okay, so the last part of my talk is about some ongoing work that I've been doing during my postdoc, which is focused on sort of our ability to apply RAD-seq across really broad and really different scales. So this is working on a group called Viburnum, which is a widespread group of about 165 species of shrubs and small trees. In the lab where I'm in now, in Michael Donahue's lab, there's been sort of decades of work on this clade. And so it's really well known from a huge number of perspectives and has been sort of a model for the application of phylogenetic comparative methods. And we're increasingly getting really close to having just a really great phylogenetic understanding of this group. So this part of the talk is work I'm doing with Michael, as well as with a lot of collaborators in his lab and previous members of his lab. So what we're trying to reconstruct is a phylogeny for the entire clade, all 165 species, as well as really dense sampling within a number of clades that correspond to really interesting evolutionary questions that are being investigated in our lab group. One of them being a focus that's been one of my main points of focus during my postdoc, which is the most rapidly diversified clade in Viburnum, which is this group that's recently entered into Central and South America in cloud forests. And we want to be able to sample really broadly across these really rapidly diversified clades, understand patterns of trait evolution within them, and then furthermore, to be able to zoom in within individual taxa or individual species pairs and study other processes that might allow us to understand, say, the genes underlying trait divergence using methods such as QTL mapping. And RAD-seq data has really sort of been ideal for this approach, and we're applying it from the scale of what might be sort of several hundreds of years divergences to a couple million years to this clade that has a crown age of about 60 million years. So previous work on Viburnum phylogeny has used a number of chloroplast markers. Now we have 9 chloroplast markers plus ITS sequenced across almost all 165 species of Viburnum. And this tree is actually really well supported. So we get really strong support for most relationships across this tree, except for these clades that have really recently and rapidly diversified. There simply just isn't enough signal in this data. And so we've sequenced RAD-seq data for 110 species in this tree. I think we're now up to 125 total. And what we find is really consistent topology that's recovered using these data, actually consistent, meaning consistent with the chloroplast tree, which I think is somewhat surprising because it's a very deep tree. You might expect just from the use of different markers that there's going to be more conflict than that. And what we find is really good resolution, especially within these clades such as the Central American clade. We've applied now a number of comparative methods trying to understand what the best way is to combine these data sets. One way we've been doing is to fix the RAD topology, which we think has much better resolution in many parts of these trees, but then to scale the data set for dating using the chloroplast data, which may be more clock-like for that purpose. And we've been using a joint estimation approach of co-inferring historical biogeography and the tree using RevBayes. We find some small differences between the analysis that we do with just the chloroplast data versus with the new RAD fixed tree. In particular, we find better support for what we thought previously was probably the case where this largely temperate plant clade, Viburnum, actually had an old world origin, originated in Southeast Asia, likely, in tropical regions of Southeast Asia. And when we reconstruct between different habitat types, we find that there's been a lot of transitions of Viburnum into a lot of these different regions, with this recent entry into South American and Central and South American cloud forests being the most rapid radiation. And that's been the focus of my research and a lot of field work we've done over the last 3 years. So as Viburnum entered into the New World around 10 million years ago, this one clade is now diversified into about 40 species. And they're distributed exclusively to these cloud forests, which are these really narrowly distributed regions that stretch from Mexico down through Bolivia. And in each of these regions, we find a really cool story where there seems to be a really similar group of Viburnum, of a mix of species of Viburnums that fit a sort of similar ecomorphological type. And that type is demonstrated by one example here of 2 species that co-occur within a region, in this case in Oaxaca, of one species that has really large, really densely pubescent leaves, and another species that has really tiny, completely hairless leaves. And one's a small tree, and the other one's a small shrub. Here's a sort of zoom in on 2 of those examples, a species pair from Oaxaca and a species pair from Chiapas. So historically, the idea for how these species are probably related to one another, and this was Michael Donahue's work from his PhD thesis, was that there was likely 2 clades that entered into this region, and they sort of left one taxon behind on each of these regions. There was a large-leafed clade and a small-leafed clade. But you could imagine alternative scenarios, the other extreme being sort of in situ divergence in each of these regions into these 2 different types, or some intermediate of the 2. And the chloroplast data just really couldn't provide any information about what was going on. Well, we applied RAD-seq data to this data set, and what we found was perfect support for a sort of ladder-like relationship moving down sequentially southward in this clade. So the first clade that comes out is in eastern Mexico, and then in what we have Oaxaca, and then Chiapas, and then Honduras, and down the line. And what we find in each of these cases is an occurrence of sister species that appears to be that large and hairy clade, the large and hairy species, and that small and glabrous species. So this really supports the idea that there's been a replicated pattern of parallel divergence along the same axis of variation in each of these different cloud forest regions that are isolated from one another. We can interrogate this further using our genomic data, and we sampled across the ranges of several of these species. And we find that these species do have a narrow hybrid zone often between them, and we detect evidence of gene flow in these morphological hybrid zones that we detect. And we ask, oh, well, what if that is actually an explanation for this pattern that we're detecting? We're just detecting recent gene flow. That's not actually the true historical phylogeny of this group. Well, we can test that by applying these ABA-BABA tests between these different regions. We can ask, does Oaxaca and Chiapas, the large leaf species between those 2 regions, appear more similar? Do they share more alleles together than expected by chance? If there's been no gene flow between the different regions, we would expect that the 2 large-leafed species, say a large-leaf species from region A, should be equally related to the large-leaf and the small-leaf species from region B. And we apply this test to a number of species pairs. In all cases, we found no significant evidence of gene flow between neighboring regions. So in contrast to all the other parts of this talk I've talked about, in this case, we don't find evidence of really widespread gene flow between these different regions. So I think this is really strong support that this in situ pattern of divergence really is real. And so now we have a bunch of future work in progress. We have a grant funded to really try to tease apart what has driven the diversification of Viburnum in this region. Why have they differentiated repeatedly into these different leaf types? We've planted a whole bunch of weather stations out in the field to try to get at the functional significance of these leaf trait differences. And we're doing a QTL mapping approach, what's called admixture mapping, to take advantage of what we think is this sort of long-term hybrid swarm that we found in one of these regions, to sample it really intensively and try to identify traits of major effect associated with hairiness and leaf size. And then to the extent we find, identify some loci of major effect within this one hybrid zone, we want to apply that across the many species pairs, these up to possibly 8 replicate pairs, to see if they all show similar divergence or segregation at these same sites that may underlie these trait differences that we've observed in parallel. So that's everything I have to talk about. I want to thank all my collaborators, which I've listed throughout the talk, and my funding sources, and thank all of you for coming. If you have any questions, please ask them. Yeah.
I'm trying to get my head around this. I understand the approach you're talking about. Does it assume that you know you have the phylogeny correct? Because otherwise, how do you distinguish between your regression? So how do you validate that assumption independently of
Yeah.
finding intervention?
Yeah, absolutely, it does. So that test, the Abba-Baba test, is a test based on a topological assumption. In the case of what we did here is we asked, well, what if that topology is wrong? If it was, then we would expect that, for example, we would find that, so the topology that we observe from our phylogenetic inference says that the big leaf and small leaf species go together. Alternatively, if the true tree was that the big leaf species go together, and this one has just integressed with a small leaf species a lot, then we would expect when we apply this test, we would, like in this case, we would find significant evidence of these two sharing more alleles with each other than with this one. And it would be not actually evidence of introgression that we're picking up, but evidence of their true shared ancestry, which would appear as introgression when our topology is incorrect. I mean, we can simply test this on different topologies, I guess, if that's what you mean, yeah. I mean, once you have a very large tree, of course, and you're trying to apply this across many different tests, there's no real simple automated way to, well, it's simple to automate the application of the test, but to interpret all of the many results under the many possible topologies, it gets quite complicated. And that's an argument for some of these more network-based approaches, but there's many limitations, sort of automated methods that try to infer a tree with network branches among them, but there's limitations of those tests as well. Mm-hmm.
And I guess I was thinking about in particular the oaks, which, as you say, are notoriously full of
Yeah.
divergence. So that assumption seems particularly problematic in that case.
Yeah.
So that's where I really started.
Yeah, I have a section that I cut from the talk, which is kind of interesting with respect to the oaks. So I mentioned that there's some of these species come out as paraphyletic. One example is Quercus fusiformis. So this is the taxon I talked about that seemed to have received a lot of gene flow in the northern part of its range from this teal taxa, and in the southern part of its range from this red taxon. And you can see that the lineage that received the most introgression comes out as sort of sister to the Mexico populations, which grouped with this more isolated Quercus brandigii. Well, what I think is going on here is that this taxon, because it received a bunch of gene flow from something further away, is that it's sort of pulled it outside of that clade that it's actually a member to. And the reason we think that is if we run this test again, this phylogenetic inference, but we remove this clade completely, so the alleles that are shared between here and here now just appear as autapomorphies within this clade, what we find is perfect support from monophyly of Quercus fusiformis, which I would suggest that this is probably the true phylogeny, or sort of phylogeny meaning, in this case, the historical relationships of these populations as they diverged across the landscape. and that this topology that we're picking up here represents the shared alleles that are going on between those two lineages. So I believe that this topology is incorrect as a representation of like lineages as cohesive units being splitting across the landscape. I mean, it's correct in terms of who's sharing more alleles, perhaps in some certain way. So it's in some sense a question of interpretation of what we want the phylogeny to represent. Oh, yeah.
Sure. So the phenotypic distance that we might measure in a plant stem or
Mm-hmm.
not always correspond to the underlying developmental distance.
Mm-hmm.
So I was wondering if you know anything that might inform your understanding of how these groups would diverge in terms of the developmental varieties.
So what do you mean by developmental distance?
Well, that one gene development seems to be very different.
Okay. Yeah. Yeah, in this case, I guess what we're interested more in measuring these phenotypic distances is the expectation that something is closer in phenotypic distance is more likely to be placing pollen in the same part of the bee. So we're asking, is it similar? We measured this distance in various ways, one being just style length difference. Other results that I didn't show was measuring distance in a multivariate morphospace that asks about color distance as well as Gallia curvature measurement. And it's all trying to get at an aspect of whether you're using bees probably at the same time in the same way. So we're not actually asking like about rates of evolution, say, but rather about a focus on expected species interactions based on trait differences is what we were trying to get at. But as I said, the morphological model was a poor fit to the data in the end anyways. So it may be that pollen transfer is relatively rare, and it just really matters whether it came from a really close relative or not is what's more important. Seems to be what the data suggest, in that the phylogenetic model fit better than the phenotypic model. Yeah.
On some of your diagrams, like the one you have up here, it looks like your progression data are kind of asymmetrical, so that the one member of one lineage is getting progressed material from a couple of others, but it's not going the other way around. Is that a common pattern? And is there a particular reason for that?
I think it's hard to tell from the structure results necessarily about how accurate it might be in terms of predicting that. In the partitioned ABBA-BABA test, you're asking specifically whether the alleles that seem to be shared between, say, these two lineages, whether they're also present in the ancestors of all of these relatives, or whether, and if it is, that would suggest that it's gone in this direction. If it's shared by these two, but it's not present in this one's close relatives, then it would suggest it came into this one from somewhere else. And so that can get at directionality more specifically. In the most of the case, when we applied those tests in the oaks, it seemed that there was bidirectional gene flow. In the case of the Cuban oak, which is this sort of most admixed one, we tried to fit some demographic models to estimate, because it's almost like 50% admixed between the southeastern clade and the Central American clade. It was really difficult to try to understand whether it was, say, historically a member of one clade that received a bunch of migrants from the other, or vice versa, or whether it was some kind of instantaneous hybrid zone from, having to do with something about Cuba. I think in the end, we can't really strongly support one or the other. We had better support for saying it was historically colonized, perhaps colonized Cuba from Central America, and has received sort of constant migration from Florida, is the model that fit best, but I don't know that I'm, I wouldn't bet my life on it necessarily. Yeah.
So, great talk, by the way.
Thanks a lot.
At the beginning of your talk, though, you pointed out that advantages of using the restriction fragment-based approach, even though you're looking at chunks of the genome. And you said that the whole genome has lots of other information that may not be useful. But I'd like to challenge you on that a little bit, right? Because one of the things that you'd like to do with these data, with integration, is to date them, right? And there are ways of dating introgressive events by looking, you were looking at SNPs here, but by looking at the sizes of
Absolutely.
the fragments. Can you do this?
Not very easily with RAD-seq data. I mean, to the extent you map RAD-seq data to a reference, you can to some extent, but it's not the best way to try to get at that data. Yeah, if you really want to look at linkage as information for the timing and source of introgression, then you're much better off trying to get really long read data, in particular, you know, sort of full genomes. I mean, I think that's great for, you know, human resequence data and Drosophila and stuff, but it's to do it for many dozens or hundreds of individuals. It's I think still quite expensive and sometimes harder data to come by. But yeah, I think that is a much more powerful approach to work around this problem of SNPs. It's much harder in many times to get at those finer timing questions.
All right, let's thank Deren.